Hi, everyone. Welcome to our 8th Tech Tutors episode. We are delighted to have you here. And today's session will be led by Dr. Richard Lamprey, and he will lead a tutorial on how do I get started with systematic reconnaissance flights for wildlife surveys.
Thank you, Netty. It's great to be here. I'm a member of the African Elephant Specialist Group, and we work with a variety of clients across Africa, NGOs, governments, donor organizations, and so on, for looking at wildlife counts, how to improve them, how to implement them, and practical implementation to provide population estimates for protected areas and that kind of thing. Okay, you'll have to forgive slightly industrial approach. As you probably noticed, I've taken Howard's slot this week. Given Howard's next week, we agreed to exchange. I have to go into the field fairly shortly for a survey in Uganda. And it's all sort of lined up for next week, weather and permits and so forth. So forgive the slightly industrial presentation. Now, I sort of went true to the title of the talk with a slight change. How do I get started with the original word was systemic reconnaissance flights, but the actual term is systematic reconnaissance flights, SRF. And that's it's sort of a distinction that we should make at this point, but I think the concept is that we're talking about the same thing, and how they have evolved, and it's a complicated subject, and how we might sort of break into this rather complex field, so. I thought I might start with some rather basic slides to do with sort of why do we actually want to do this? Why do we want to fly across the landscape, doing a reconnaissance of the landscape for enumerating animals and other variables? Well, in Africa, as in other areas, Australia, parts of the United States, we have very large areas of rangelands, occupied by some people, wildlife, livestock, very large areas that we wish to know what are the wildlife populations in these areas, what are the other variables that might affect the distribution of these wildlife, these different species. And as you might stare out the window, you say, how on earth do I get started with that? It's such a big area. And so people have been getting to grips with this over the last 60 years or so. One of the first things they realized was that randomly flying across the landscape to get an understanding was not generating very, very useful information. They weren't sure about the accuracy of the information. They weren't sure if there were sort of biases involved, if they'd even covered the extent of the wildlife area. Sort of through the 1950s and early 60s, they muddled along with total counts, which is something I'm going to come on to very briefly in the next couple of slides. But really, the starting point for the systematic reconnaissance flight was a study done in Canada in 1963 by Ron Skoog and Ray Sinif on caribou. And they were tasked with finding out how many caribou there were over a very, very large range area of caribou in northern Canada. And they were the first to come up with a systematic idea. They said, well, there is no way that we can fly our little planes over this whole area and try to count all of those caribou, hundreds of thousands of them. What we have to do is roughly find out which sort of area that they are, what is the extent of that area, and then design some samples. We sample bits of that area in terms of the terrain. We understand how much terrain we're going to look at. And if we can understand that, and we can then count the caribou within that those blocks of terrain, we might be able to come up with an estimate for the whole area. And this is what they did, flying in little two-seater airplanes. They divided it all up into blocks. They randomized the blocks so that they were not biasing it in any way. They weren't putting all their blocks in the places where all the caribou were, because there are many here, a few there, but they all added up to a total population. And this was the very first systematic reconnaissance flight that was done, as far as I can make out. And so it was a landmark study, but at the same time, far across on the other side of the world, the Serengeti Research Institute was starting up, and scientists were coming to the Serengeti to understand a very similar problem, which was where are the wildebeest and where do they go at different times of year? And remember, at that time, in the sort of mid-60s, nobody actually knew the route taken by the Serengeti wildebeest migration. So a number of sort of initial surveys were done to try to determine where they were, what was the extent of our survey area, so we could work it out. But in the end, they decided the survey area was essentially the whole Serengeti and up into Masai Mara. And they started to design a program in which they would fly lines across the landscape to and fro, separated by a certain distance. And given that they only had sort of very small planes, 2-seaters, 4-seaters, the idea was to have an observer in the back looking out of the window to left and right, and to determine roughly how many wildebeest they were seeing on each side. And once they had done that, they could start to further enumerate a survey area. Now, they did this for every month of the year. I think it was through 1968, 69, flying those small planes in blocks across the Serengeti, drawing up, sort of flying on a grid system, with people looking out the window. They weren't necessarily counting properly. In fact, I've got some of the old transcripts. They simply said, well, there's 5,000 heads and 70,000 tails. They had a sense of humor about it because there were just so many. But in the end, they built up a picture of the wildebeest distributions so that they could then start to understand, if they're going to count them, what's the best place to count them, at which time of year. This is why we have this rather strange name, systematic reconnaissance flight. What's a reconnaissance? It's going out there to sort of find out. It doesn't necessarily imply the word count, but it has its roots back in the Serengeti when they were trying to work out where wildebeest were at different times of year, in what densities, so that they could then concentrate their efforts on doing a proper count as and when they knew they were concentrated in certain areas and easier to count. Now, as that project moved forward, there was a sort of key study done, a key conference held in Kenya in 1969 to try to work out a better way to do the actual counting, rather than just looking out the window and estimating. So at that conference, there was invited a Scottish statistician by the name of Mr. Jolly. And he's never ever presented with his first name. He's just called Jolly. And he came up, he was a very eminent statistician from Edinburgh who came up with the statistical tool that could be used to count animals over large areas. And he designed this statistical process, which persists to this day, and we'll go into it a little bit later on. And it was only then that they could actually do counting. But the term systematic reconnaissance flight first appears in 1975 in a conference on rangelands in Bamako, Mali, where the various experts got together and seemed to come up with that name. And it stuck for forevermore. And so generally speaking, it's how you fly in a systematic way across the landscape in order to enumerate wildlife, livestock, and other environmental variables in such a way that it's done usually according to a grid system, so that you can analyze with respect to a number of variables per grid cell, and do sort of various spatial and multivariate analyses on why the animals happen to be there at that time. Is it something to do with human settlement or distribution of water sources and so on? So this is how the SRF came to be. And if you look, for example, at the Serengeti, you can see that it really is a challenge. Carpets and carpets of wildebeest. How on earth do you count those? You can't count them all. You can only count a sample of them. And if you do it in the systematic way, then you can map them out right across the Serengeti ecosystem and see where they are at different times of year. And in respect also of other species that occur within the same grid cells. Sorry, and not only that, the rangelands are not just a huge extensive grasslands covered with wildebeest. You have very, very varied habitats across East Africa, where visibility is different. There is thick bush and woodland cover, very green landscapes. This one of Queen Elizabeth National Park in Uganda, and methods that you simply cannot find every elephant in this landscape. You can only find a sample of them. And I just draw your attention to certain classic texts to look at this, designed sort of through the 1970s and then 1980s, which are critical reading even now with all our new modern technologies. The essentials have not changed since then. We might be using better equipment. We might be using fancy cameras. We might be using all kinds of navigation aids that they never had then. But the principles are essentially the same as were worked on at that point and agreed upon. I'm sure you've all heard of Counting Animals by Mike Norton Griffiths, which summarized the state of knowledge up till 1978, brought together the concepts that have been developed in the Serengeti into, and as with the statistical processes designed by Mr. Jolly, into this classic text about how to design aerial surveys. There were many questions remained. There were very vigorous debates, culminating in a meeting in 1981 in Nairobi, low-level aerial survey techniques, where they thrashed out all kinds of issues, one of which was Mr. Jolly's assertion that you should simply randomize the transects across your area, all flying in one direction, but if you imagine a baseline running through, those transects could occur at any point, a random point along that baseline. And it was decided at that point that actually the systematic way, where it was generally the same spatial distance between transects, generate a grid system, could equally use the Jolly 2 technique. It was equally applicable, and all of these details were thrashed out at this survey, low-level aerial survey techniques. At the same time, in a completely different landscape, they were looking at things in a different way, or did different sort of set of problems in Alaska, how to enumerate their moose populations. And there is a classic text, Institute of Arctic Biology, I think 1986, by Gassaway et al., that incorporated a lot of the processes of Jolly, but also a range of other techniques as well for enumerating moose, which don't occur in these massive herds that we have in the Serengeti. These are all classic texts for those who might be interested. Since then, there have been various sort of evolutions, but none has none of these have done other than sort of polish up the various aspects with new technologies, and more rigorous procedures, but the basics still remain. The same for the systematic reconnaissance flight. Now, I'll talk a little bit about total counts. This is where you have an area, a protected area, and you simply want to know how many, and let's use an example, how many elephants there are. And so you're going to set out to count absolutely everything. And these counts do occur. They occur every 4 or 5 years in Kenya to estimate the elephants in the 35,000 square kilometer Tsavo ecosystem. And lots of aircraft get together, people come together, counting teams, they divide the area up into blocks, and they do their counting. They're used when a total number is needed without consideration of a margin of error of the count. There is no margin error. You're simply setting out to count everything. You counted it in blocks. You divide the area into counting blocks, e.g. for a convenient day's flying. You count all the target animals in each block, and you add up all the numbers, and you come up with a population estimate. And since you've counted everything, or as far as you understand, you've counted everything, then there is no margin of error, because you're not creating a sample with which to sort of compare the estimates for each sample. The advantages can be done with relatively untrained crew, but with good eyesight. People can come in who are interested to participate, fly in a plane for hours, count to left and right, and they slowly assemble this big database on how many elephants in each block, and then it's all added up, plotted in the GIS system, and you have your distributions as well. The disadvantage is the whole area is covered in narrowly spaced transects, hence many hours of flying, and hence expensive. This is, for example, the total counting blocks for Murchison Falls National Park in Uganda, which we do as a total count probably every 20 years. That's when you get this sort of kind of money to do this in other countries. But you count each block, and you come up with your overall population estimate. And here is a sort of classic counting pattern: total count, transect space 1 to 1.5 kilometers apart, plane going up and down, people looking to left and right, marking down onto their data sheets or talking into their tape recorders and recording where those animals are. Now, that's expensive. And, you know, to get 10 planes assembled in Tsavo for 10 flying days is a very expensive operation, and not everyone can afford it. And not only that, it's no good for, if you're going to be hunting down the elephants, then to try and find all of the Thompson's gazelle as well. You'll simply tangle yourself up and fly around in circles. So it's good for big conspicuous species, but if you're going to try and count a range of species from big to small, you're going to be circling around forever. And so when it comes to the systematic reconnaissance flight, we only sample that a part of that. So we don't have to worry about the rest. We just have to worry about what's in a sample. Assume that if we count all the animals in a known fraction of the survey area, then we can extrapolate to obtain a full population estimate. The concept is very, very simple. We count in sample units that are representative of the habitats of the survey area. So, for example, if we cover it with regularly spaced transects, we'll know that we will get the woodlands in the proportion that the woodlands exist in the survey area, the grasslands in the proportion that they represent. And we know that we are sampling those representative habitats, and we are not biasing towards the woodlands, which may be in one corner, or towards the grasslands, which are in the other side of the area. We're looking at the whole area in a systematic way. We add up the physical area of the samples and the total animals counted. And then the concept beyond that is actually quite simple. If we counted 50% of the survey area, then we assume we counted 50% of the animals. Now, what are these sample units? Generally, these sample units are strips of terrain. As we saw earlier, they don't have to be. Mr. Skoog and Mr. Sinif in Canada used blocks. It's a very similar process. They certainly did. They didn't count everything. They counted their sample blocks. But in the systematic reconnaissance flights, the sample units are strips of terrain. These counting areas are defined by markers set up on the aircraft, and rear seat observers in the aircraft count animals that occur within these strip transects to the left and the right of the aircraft. So they are strips of terrain, and the important thing is you know how big those strips are, because if you counted 50% of the terrain, then and you're confident about that, then you know that if, for example, you counted 50 giraffe in 50% of the terrain, then you might be looking at 100 giraffe in 100% of the terrain. It's as simple as that. And generally speaking, it's set up so that you would not biasing the transects in any way. Either you can set them up randomly as the old-fashioned way of Mr. Jolly, or systematically, but you are not concentrating them in the areas that you know more, and then using that to extrapolate to the whole survey area. It's a systematic process. The rear seat observers in the aircraft count animals that occur within these strips. Now, along the way, if you're interested in this topic, you will come across two very contrasting types of aerial sampling. Now, I don't know if you can see at the top of the screen, you can see the aircraft on the left. We're looking at two strips, one side of the plane and one the other side of the plane, while the one on the right is flying along a line. And let's deal with the one on the left first. We've described roughly how this is done. And the plane flies along a transect, the strips are to left and right. And generally speaking, it's set up to define a strip width of about 150 meters on each side of the aircraft. And that's been done through some long experimentation in, particularly in Kenya, in the late 1960s and through the 1970s. What is the best strip width? And people, obviously, if you want a bigger sample, just increase the strip width. And so there was a lot of work done early on in implementing early SRFs with very wide strip widths, because they thought, well, it's really expensive to keep this plane up in the air. Let's increase our sample by increasing the strip width. And this is what they did, particularly through a series of surveys in Zambia. And what they found, they got one of the classic SRF technicians in, Graham Corley, from Australia, to review the results of these early SRFs. And what they found was that the number of elephants in the area declined in response to the strip width. You know, the bigger the strip width, the more elephants. The smaller the strip width, the more elephants. The bigger the strip width, the fewer the elephants. And what they realized was that elephants out towards the edge of the strip were simply being missed. The observers in the back could not scan in and out on a 400, 600 meter strip. Plane is moving forward too fast for them to do that. And so the declining Zambia or Luangwa elephant population was not a result of a declining population. It was a result of increasing the strip width in order they got a better sample. So that 150 up to 200 is pretty critical if you're going to have people in the back of the plane trying to count animals in the terrain that is passing. Now, a different way to do this, which you will come across particularly in America, is what's called the line transect, where you fly the aircraft along the line and you measure the distance to the groups of animals to left and right. And when you do that, it's a thing called distance sampling to determine your population. Now, the idea behind this is you don't waste information. In the classic strip sample, you count everything that's in the strip and you omit what's on the outside. You do not count it. You might note it, but it's not part of your count. In the line transect, they said, well, it's obviously expensive to keep a plane up in the air. Let's count everything. And then come up with some kind of a function that describes this drop-off in observation as it gets further away. And you'll come across this a lot in America, particularly where they have helicopters, which are cheaper to run. And you can actually slow the helicopter down and try and work out how far that animal is from you. It's hard to do in a fixed-wing aeroplane. It has to sort of barrel across the countryside at 170 kilometers an hour. But with a slower flying plane or a helicopter, you might have a chance at measuring that line. And this is the line transect technique is very prevalent, very much used, particularly for single species or double species counts in difficult terrain in America, in the Rocky Mountains, or in areas of thick cover. And it has been tried in Africa. It's not part of the systematic reconnaissance flight technique, but it is worth dwelling on it just a little bit. There are assumptions to the line. That on this line, 100% of the animals are counted. And the animals are fixed in their location and do not move. In other words, they don't go from sort of near the plane to far from the plane, which is a little bit problematic if they're running as fast as they can from the plane. And that the distance of the animals is accurately measured. Now, you can also try line transects on the ground. This is one in Masai Mara, and you're dependent on this detection distance, which is, it's called a hazard function. All kinds of different hazard functions for different animals. How far do they have to be from you before they become invisible? And you calculate this function for different animal species. There's lots of different ways of doing it, and there are computer programs that will do it. And of course, for ground counts where you don't have a plane and you're in the sort of environment where you can measure distance and so on, distance measuring is a very important function of enumerating wildlife populations. But it's quite hard to set up in a plane. You've got to set up all those additional rods and markers for defining what is 100 meters, what's 200 meters, what's 300 meters from the plane. And you've got to have people sitting in the back of the plane trying to work out, did they see the eland or the buffalo or the giraffe within that first set, second set, third set, fourth set, going way out, far distance. Wires on this is in South Africa in Kruger National Park. And so it becomes more and more complicated for those who are wanting to enumerate a multiple range of species, particularly in African environments, and where the complications of setting up. And so it has been tried, but the results have not been satisfactory in Africa. And it is a sampling technique that you will be, you may, if you're interested in the subject, will be aware of, and it is used extensively, as I say, in America and sometimes in Australia. So the strip trans, the basis of the SRF method, all the animals within the strip are counted. Animals outside the strip are ignored, and the area of terrain covered by the strips is known. So let's imagine we have a population of 100 animals. Here they all are, nice square survey area. And you have a series of transects across the area, all equal sized. Each one has 10 transects across this area. And there's 100 animals in this population. Now, this is a sample size of 50%. And in this 50% sample, we count 48 animals. And our population estimate is 2 times 50%. It's 96 animals. So just to illustrate that very simple concept. Now, one of the things you will notice here is that these animals are pretty well randomly distributed across the landscape. And in such a rather rare situation, that you have such a perfect counting block with random animals in them, a systematic sample generates very, very predictable results. Here, just by eliminating 50% of your flying, you have a population of estimate of 96 animals and not 100. Here, we've eliminated, we've gone down to a 20% sample. We count 16 animals, and our estimate is 80 animals. So you see, precision is beginning to drop off, but this is 1/5 of the cost of doing it, the whole thing. So this is the great advantage of sample counts, is that we can work up an estimate by reducing the sample, by having a lower sample than you would if you were trying to do the whole thing. This is from the classic text, basics of obtaining a sample estimate from Meitner and Griffiths. Here they have sampled this transect, counted 14 animals, this one with 9, this one with 10, and this one with 9. And this goes into your estimate as an estimate of 105 animals in this sample. And by looking at the variability between these sample units, we come up with a margin of error, like 95% confidence interval of, say, 87 to 123 animals. So here you'll see the animals are more clustered, but this is still generating a very useful result, but of course this is purely conceptual. Now, here, imagine this area, we have the river running through it, okay? Here we have a total wildlife count of 115. There are 115 animals in here. The total in the sample is 49. And this is about I think this is a 50% sample, as I sketched it out recently. We might be estimating about 98 animals out of this population of 115. That's our estimate. But one thing you will notice is the numbers associated with each transect are highly variable. In this one, we have 0, this one 9, this one 37, and this one 3. And if we look at the variation between these samples, the margin of error, what we calculate is a thing called the standard error, is very, very high. The confidence intervals on your estimate will be very, very high. And you will not really be sure exactly where that estimate is. It'll be somewhere between a very wide range of possible estimates. Let's do it the other way. We cross that, river at perpendicular. And here you see very different type of estimates. In our sample, we have 63. Assuming this is a 50% sample, we would estimate 123, 126, and not 115. But our margin of error would be much smaller. We have counts of 13, 14, 20, and 16. And consequently, we would have a much smaller standard error, and we'd be able to be a lot more confident about our estimate. And this is really critical when you're trying to compare surveys, for example, one done of the same area 10 years ago with this one. You don't want to have too much variability because you want to identify a trend. You know, if you say, well, it's gone up, but we can't be absolutely sure because the margins are so big, does that really help the manager? So the design of an SRF is absolutely critical. in terms of how you design the transects across the landscape. Now, how do you actually define these samples? In the plane, you have wing counting markers attached to the wing struts of the plane. And this is just a graphic demonstration of how this thing looks. And here we have a group of sheep going under a tree, and you're counting everything between those markers. And you're setting up your aircraft very, very, very carefully, and measuring on the ground, extrapolating to your strip width at a certain flying height, using standard methods, this one again from Counting Animals, of how you set up a strip width of 150 meters when flying at the flying height that you want. Here are some markers on a plane. This is, you can see them here on the wing strut. Here they are on the wing strut of this aircraft, set up so that the people sitting in the back looking out will have, at the set flying height, you will have a strip width on the ground of somewhere 150 to 200 meters. And here they are setting up their sample designs, making sure that those markers are in the right place on the wing to define that strip width. It's called the pre-flight calibration of the markers. During flight, the observers, rear seat observers, are looking out through the markers. You can see a marker here, and there'll be one further in, and counting the animals within that strip. In this instance, they've also got the camera attached so that when there's a big herd passing between the markers, they don't necessarily need to count it, or they make an estimate and then take a photograph for later analysis in the lab. She's got a camera trigger in her finger so that when she's counting, she's talking into the microphone, the estimate that she can actually trigger the camera as well. And as she's looking forward here, between the markers is a group of elephants. Now, when I was talking about a certain flying height, what is that certain flying height? Well, again, those studies in Kenya showed that if you're going to have a multiple range of species from elephants down to example to Grant's gazelle or something like that, the observer can't really see Grant's gazelle or Thompson's gazelle if you are up at 500-600 feet. They can only see those gazelle if you're down quite low at 300 or 350 feet. And therefore, the general standard for setting up an SRF and for your flying height is around about 300 feet or 350 feet. You would need to decide. Just in some cases, it's gone to 400. The Kenya government ran its entire SRF program at 400 because it was flying twin engine planes, which if they have any kind of a big problem, 300 is not good enough. It's not safe. And in fact, the donors call for an additional 100 feet. They're going to fly everything at 400 feet. But generally speaking, 300, 350 is the standard, with 150 meter strip width each side, and you go up to 400 under certain circumstances. Now, obviously, the higher you go, the bigger your strip width. So in this instance, we're looking at the initial one of 400 feet at 150 meters each side. You double that to 800 feet, and you have 300 meters. So the bigger your strip width, or the higher you fly, the bigger your strip width. And consequently, the higher your sample size. And as we've seen, the sample size is actually critical. We have to understand what that is before we can extrapolate to the whole population. If you don't know what you sampled in terms of the percentage, you can't get an accurate figure afterwards of the number of animals. So flying height is critical to this whole thing. And to do that, once you've set your rods up in the hangar on the ground, you then fly your plane over ground markers. It's called the strip width calibration, to determine the size of your left strip and right strip, whether it's sort of within the margin that you want. Might be sort of between 130 and 170 meters, but you might be aiming for 150. But you have to know what it is. You fly the plane over those markers at certain heights. The observers in the back count the number of markers that they see, and you then work out what is your strip width for the given flying height. And you come up with a regression that's something like this. This is your radar altimeter. This gives you your height above the ground, tells the pilot how high the plane is above the ground. And you have to be very careful. You distinguish that from how high above sea level. That's completely different thing and meaningless in this situation. How high are you above the ground? And this is your combined strip width, both left and right, for that altitude. Yeah, and this is a survey that we were doing in Rwanda. And we were flying then at, I think we were aiming for 400 feet. And we had a combined strip width at 400 feet of about 280 meters, 140 meters strip width each side. But given that this was into the Akagera Valley, where you're coming off big hills and going down into the bottoms of the valley, we had to have a very big range. So we tested it right up to the very tops, even knowing that if you came off the top of a hill at 1,200 feet, you probably wouldn't see much apart from elephants, but at least we remained safe. So in hilly environments, it's very important to have this radar altimeter calibration. And if you aim to cover the hilltops at about 300 feet, you're not going to get down into the valley. The plane will not climb up the other side. It will impact the valley wall on the other side, which is what you don't want. And you have to take compromises along the way of increasing and decreasing that flying height. Hence, you need a fairly wide-ranging strip width calibration if you are in hilly country. So looking back again at a typical census zone with the river running through, a whole group of animals, different species, different colors, yellow ones living up on the hills, red ones on the swamps down at the bottom, brown ones in the shallow lands here, and water-dependent species along the river. Designing your strip width, your sampling procedure across that gradient, across that watercourse where you have those congregations. And this is how it might look with that double strip width that we have on each side of the plane. In this instance, there are 445 animals in this. This sample is a sample of 13.3%. 59 animals are counted in these little gray lines, and our population estimate is 442, which is very close to that estimate, the total number that we know, which is 445. Of course, this is all conceptual, but it's to illustrate the principle of the SRF. Now, Jolly 2 analysis is very simple. I'm not going to deal with the derivation of the sampling error at this point. But Z is the area sampled for each transect. Y is the number of animals enumerated along the transect, and so R is the animals, which is total number, divided by the total area sample, the average density. And what you do is simply multiply that average density by the size of your survey area, and you get your population estimate Y. And the great thing about doing all this in a grid system is that you can integrate this data with other data, climate, soil types, population density, and infrastructure in a GIS system for building up a picture of the resources of your survey area and how, for example, your livestock or your wildlife might be influenced by the availability or otherwise of those resources during the time of the survey. It can be done also at different times of year to look at seasonal effects. Well, I'm going to talk about bias and the problem of animal detectability. It's well known that RSOs do not detect all animals of the target species. How do we know? Some animals are not available for detection. For example, marine animals, they might be underwater. Terrestrial animals are under tree canopies or hidden from view. However, when you look at available animals, those that are there to be viewed, many experiments show that rear seat observers miss many animals. The job of a rear seat observer is long, boring. They sit in planes in uncomfortable situations, often feeling a little bit exposed to danger, to noise, vibration, to heat. And in this difficult environment, they're expected to count animals to left and right in an accurate way. But they're only human, you know, and after 2 hours or 3 hours of this type of work, they start to lose concentration. This is natural. So not only that, there might be some factors as well. You know, the animals are particularly sort of cryptic. They blend into their backgrounds. They might be there to spot, but if your eye scans over them, it might not see them. Group sizes, if it's a small group, you may not pick it up. If it's a huge group of elephants, you might, or you're very likely to. It's just going to wake you up. Might be all kinds of things, animals in multi-species groups. How do you disentangle them? You know, you've only literally got each group in view for about 7 seconds before that plane has moved on. And you're expected to enumerate those and to do it accurately. Behavior to the aircraft of those groups of animals. They might run, they might split off into groups, they might charge here and there. And also the lighting conditions. You might be flying early in the morning so that you get them while they're out in the open and not hiding under trees, or late in the evening. But you're looking into the sun in one direction and down sun the other direction. All of these things affect detectability and create a bias. You didn't see them all. Now, our experiments show, we did an experiment in Tsavo in 2017, in which we had the rear seat observers and we had the cameras running. And we recorded continuously recorded the strips that the observers saw. Now, this is different from an observer who sees a group of elephants and takes a picture. What happens to the elephants that he did not see? He didn't take a picture, so we don't know. This exercise photographed the entire strip that the observers were looking at. Now, of course, a lot of that strip doesn't have any animals in. You know, it goes on and on over dry countries, no animals, but the cameras just click away. And in the end of that exercise, the core area of Tsavo, which is about 10,000 square kilometers, where most of the elephants are, we obtained about 80,000 pictures. And for the other blocks, we obtained about another 80,000. So 160,000 aerial images were taken of Tsavo of the same strip that the observers were looking at. And what we found at the end of that, and we interpreted 160,000 pictures in 9 months. We had 12 interpreters and data entry specialists working for nearly 9 months to look through 160,000 pictures. We found that the observers were missing about 60 to 70% of the antelopes, 60% of the giraffe, about 30% of the elephants, about 45% of the zebra. So it suddenly became acutely obvious that these counts that had been done for years and years in different countries in Eastern and Southern Africa had missed so much that that was there, but simply hadn't been detected. Now, I'm going to get onto this a little bit later on, but just the fact is to show that observers miss many, and that you have to be very careful. And the more you ask them to count, the more species you ask them to count, the more difficult species, then you start to get these errors, these biases creeping in that really can distort an entire trend for an entire country if it's done on and on in such a way that we really aren't sure how many animals they're missing. Now, not only that, you have precision, issues of precision, variability in the estimates. Now, I showed you how those different transects, all the numbers, if you have high variability in the numbers, lots of zeros, and then lots of hundreds and 50s and so on, you have a difficulty in the precision, your margin of error. And this is an example from Savo over whatever it is, it's about 30 years of sampling. And you can see the error bars on this estimate are very high. Look at the red one, the elephants. Those numbers are up and down, up and down. And if you were to say, well, are the numbers in Savo increasing or decreasing? You'd actually be quite challenged to say which way they are going. What is the trend in that? As things got better, the counts improved. You got this more regular estimation and slightly improved precision. But in the time when this was sort of run by a country program, a national program, where people really were asked to count week after week, month after month, and then go to other areas, you could see that the estimates were really beginning to come apart. Now, the bottom line is, the blue line is oryx. You can see possibly a decline from the 1980s into the 1990s, from about 7,000 oryx in 1977. We really don't know, because at that somewhere along the line they started to fly bigger twin-engined airplanes instead of the normal single aircraft engines, single-engine aircraft. Two engines, one outside the window to the left, one outside the window to the right. How easy was it to count? And with these droning engines going on for hours, is this an artifact of the data set that we just have errors, we have biases? It's very, very hard to tell. One critical point is, at this point, they went back to single engine planes. And at this point, they put cameras on the plane, or we put cameras on the plane. This is the estimate for oryx in 2017. It's about 9,000, up from about 1,000 that was being counted in the early 2000s. Oryx are very cryptic, very easy to miss, but when you have an air photo, they are right there looking at you, and you count them. Now, what happens if you try to reduce your sample intensity to cut costs? You only have a certain amount of money to do this. You can only keep the plane in the air for so long, because no one's paying for it to be in the air any longer than that. This is the variability in estimates with the sample intensity in Queen Elizabeth National Park in Uganda in one block, which is actually one of the highest density blocks for elephant in the world. It's 1,000 square kilometers, and elephants are at 3.3 elephants per square kilometer. And if you look at your bottom end of the sampling intensity, often what's typically done across East Africa, and you simply say, okay, we're going to randomize our transects out of a number of possible bunches, and we will get a range of possible answers as to how many elephants there are. And what we will find is that if we take, for example, a 10% survey, which might be, I think, about sort of 5-kilometer transect spacing, absolutely standard for a systematic reconnaissance flight, we get elephant estimates ranging from 2,000 up to 7,000. And so we've done the flying, we spent the money, and we have an estimate of 2,000. Last time it was flown, it was an estimate of 5,000. Is that a decline, or is that just due to sampling error? That we are not sampling enough. And what we did, we flew a very intensive sample, just about the highest that we could do, with 1 kilometer transect spacing. And this is what we got here. In this area is about 3,700 square kilometers, 3 elephants in this block. You can simulate elephant populations along a transect to see how this tails off. And what this means is that the more transects you do, the higher the sampling intensity, down it goes till this line here, which is your 95% confidence interval, declines to the point where you can be pretty certain that there's 3,700 elephants there. So anything lower than that creates a problem. And yet so many of the surveys done in Africa are hovering around here, where you just decide to pick whichever line of set of transects you're going to sample out of your total number of possible transects. And you might get this kind of scatter. And so relative margin of error, which is basically the 95% confidence interval divided by your mean, is, generally speaking, everyone is very, very happy if we can get that down to about 20%. In fact, the guidelines for elephant counting say that's really what we should aim for. But in generally speaking, sample intensity is low sample intensity. All you can afford is getting you an RME of 60%. You're not really in a position to determine trends accurately. And what we found, that only approaching 30% just, and at 50% likely, that you could spot a change in the population of 40% with absolute statistical clarity. And that's a big change. Say, well, to your manager, I'm afraid I can't, it's 40%, but I'm not absolutely sure because it might be sampling error. He needs to know a bit more than that. And for that, you need to fly more, and you need to increase your sample intensity. And as you see, as you know, as we've seen, the 150 meters each side, for people looking out the window, you can't widen that up to get any more. And so you're tied in to this slight sort of paradox of how much sampling is needed to demonstrate your trend, and to be able to tell with confidence that there is something happening to a wildlife population, and it's not just your sampling error. Now, detection bias, this is precision we've spoken about, which is how precise do you think your estimate might be, plus or minus a certain amount. But of course, your estimate might be completely off because you're missing all of the unavailable animals. And how much you'd be doing this? Well, in this picture, we have just a lovely piece of Savo bushland, and under that tree are a whole lot of zebra and a whole lot of buffalo. And your observer will not see those. They're deep in shadow, deep in contrast. His eye will be over this scene probably in about 3 seconds, and he will not see those animals. Similarly, in this situation, hidden elephants under a tree canopy. This has got 14 elephants in it in this picture, but no one's going to see them. Only if you can freeze the scene. Here you see a whole group of elephants and a whole lot of them huddled together in the shade of this doom palm on the banks of the Galana River. So this is why observers miss 30, 40, 50%. You're asking too much of them. But over the last sort of 20 years, there have been more standards designed to try to sort out some of these issues by standardizing on techniques, defining your flying heights and strip widths, your navigation parameters, your crew duties, exactly what they will do. Are they just observing and calling to somebody else to record, or are they both observing and recording and taking photographs of big herds? How the data are recorded in the plane, the lengths of the transects, and checking for biases between observers. There's a range of statistical tools for that. And this is a case example from Savo Cord, March 2017. You see a line across here where, according to the standards, we should turn the aircraft around. The guys have been on the line for 30 minutes, and they will be beginning to phase out in their counting. And we should not ask them to do more without taking a little break. And so we go up the line, we turn around at the end, they have a maybe 3 or 4 minute break as the aircraft turns around, have a drink of water. It's 38 Celsius inside the plane. And we go down another line. And so they get the refreshment at the end of each line. And we break it into each long transect into 2. So this is a sort of practical example of breaking a transect for observer comforts according to the elephant counting standards, the primary one of which is called CITES Mike version 3, which was designed in 2019. Now, I've put this here in order to show you, it's probably slightly in the wrong place, but it's on the basis of the SRFs, and sort of by 1977, the Kenya government was censusing the whole of Kenya, the whole rangeland area of Kenya, with small planes, with observers in the back, doing systematic reconnaissance flights, and coming up with estimates. Usually every 10 years, they would do a livestock count for the whole country that sort of fed into sort of agricultural development, economics, country priorities for livestock and agriculture. And so this was being done across the whole country. But, you know, as you can see, this, if I go back here, this elephant line is from one counting block, Taita Taveta in Kenya. And you can see that really there were errors that were coming in that where we need to sort of think about some new ideas, bring in some new technology into this process. Sort of by the turn of the century, we were getting some digital cameras. One of the main things that was to sort of standardize on teamwork, really forge strong counting teams, treat them well, look after them in the field, make sure that, you know, in this arduous task, they're comfortable and they can get on with the job of counting. This was one very good feature of the Kenya government's Department of Resource and Remote Sensing surveys, and they forged that team together for years and years, and had some absolutely expert people. But it was, as you can imagine, a countrywide survey with small planes is very expensive. You had donors who were prepared to, the World Bank, you know, the Canadian government, who always had an interest in counting and supporting counts, were supporting that. So you had to, this particular team, now that's me on the left, were supported by European Union to systematically count the protected areas of Uganda. About every 3 years we did a count, but according to the traditional ways. But we have evolving technologies, and Howard is going to go into this in more detail at the next meeting. Things have been made easier by a whole lot of things. First of all, since the early days, you can feed all of your transects into a GPS, all of your subunits for your subsequent mapping. That's fed to the pilot or the pilot screen, and it makes the navigation much easier. You don't have to spend hours poring over maps, trying to work out your bearings and your distances, as I used to do in the old days. Sometimes getting lost, wondering where they were. That doesn't really happen anymore. You can zoom in on this. This is our flight lines. This is a Lake Mburu in Uganda, and how it presents to the pilot when we're doing a count, so we know where we are, which subunit we're in, and all very useful for navigation. You can even use other tools. For example, in Uganda, we use digital elevation models for maintaining height. In this instance, we're going to camera counts, and I'll talk a little bit about that. And we're flying at 600 feet above sea level, above ground level, because we're not limited to a person who can see a gazelle at 300 feet and not at 600 feet. We have camera systems that are good at picking up a gazelle or an oribi or a small animal at 600 feet. All of this feeds into the GPS unit so that we know pretty well where we are. And starting in 2015, we started to put cameras in because the camera systems had reached the point, megapixels and the storage capacity to be able to put them out of a window and instead of having observers, to actually get rid of the observers completely and photograph the whole thing. In this instance, we had a donor who was so concerned about health and safety, they simply couldn't understand why anybody would want to be in the back of the plane behind a single engine. You know, for them, they needed many engines, you know, in case one of them stopped. And so they said, no observers. And so we put cameras instead and photographed the scene below, the strip width, gathered thousands and thousands of images. This was Murchison Falls National Park, Uganda. Or you can put in cameras through the hole in the floor of a smaller plane, Cessna 182. Howard is going to demonstrate some very neat arrangements for fitting cameras to wing struts of a plane, which means that we don't have to be limited to planes with holes in the floor or special opening windows, but whatever plane might be available in that country. And we have diagrammatic image footprint string. This is left side. This is overlapping strips. The cameras are orientated at 45 degrees. And we have overlapping strips to capture an entire strip. This is how a strip looks like when it's plotted onto Google Earth. You have the line of flight. And these are two subsequent pictures. You can see a minivan driving along the road here. 2 seconds later, the next picture, the van has progressed and is almost under the tree. And under these bushes, you have Uganda cob, you have hartebeest, and you have buffalo. So you can freeze this on your screen. It's not there for 5 seconds. It's there for as long as you want it to be. You can look into the bushes. You can look under the trees. You can look out into the grasslands. You can spend 10 minutes on this picture if you want. Of course, you know, the survey will take longer because somebody in the lab is doing this, but you capture what is out there. And this is why we're seeing, you know, that observers are missing a lot, because how can they possibly absorb what is happening in this scene when it is so busy? And then this was early training of image interpretation. This was actually for Savo between Kenya and Uganda teams of how to interpret the images, how to enter these data into a database for analysis using the standard Jolly 2 method. Coming up with distributions that are needed by the donor. Where are the Uganda cob in Murchison Falls? So that they can plan accordingly. What are the numbers? Is it going up or down? Now, having people like this, you know, who have come from university. We've got to train them up. It's a lot of work, and it's also not very easy work to have people staring at photographs day after day for 9 months, thousands and thousands of them. It's a relatively slow process, and it lengthens the time of a survey. For example, if you have rear seat observers, you know, when they're in their comfortable hotel, they get their tape recorders, they download what they saw today onto data sheets, and it goes straight in. If you do it this way, then it's literally thousands of pictures, and it will take months and months to look at every single picture. This is the joy of holding a frame for 10 minutes while you accurately count everything, but it lengthens the time of the process. How can we make that go faster? Well, we're going through a series of experiments at the moment in which we're training computers to identify the animals. And here we have, it's called annotation, which we feed this data into the computer. This is an elephant, and you keep presenting thousands and thousands of elephants to the computer system in what's called a neural network. And the computer starts to learn what an elephant shape and form is. Now you need thousands of these to train a computer, and you need them in the sort of the form in which they derive from an aerial survey. It's all very well to fly, for example, a drone up or above a herd of elephants and photograph it from all different angles. But in the context of how it's gathered in a way that is accepted by the scientific community, which is sample strips to each side of the aircraft, until we have a fully agreed replacement for Jolly, this is how it would be. And this is how elephants appear in that sample of strip to the camera and to an observer, of course. So train the computer in how you see. And of course, you know, some elephants are extremely camouflaged, as we saw earlier, under a tree. It might even just be a representation of a couple of tusks protruding from underneath the tree canopy. A human, you will recognize it. Those are tusks. But you then have to train a computer system that 2 tusks coming from under a tree equals an elephant, and you don't have the rest of it. So training is a hugely complicated process, but it's advancing very rapidly. We've been working with a group in Seattle who have designed the software for the training so that the interpreters can actually do the annotation and the system learns at the same time. And the system comes back to you with an identification. That is a cob, that's an elephant. And it can save time. And in the end, what you can do is eventually filter it out to the point where you say, okay, this computer knows that 60, 70% of these photographs have got nothing in. There is nothing there that a person needs to look at. But 30% have something. We may not be absolutely right. We may not be certain it's the difference between a hartebeest and an impala, but a human can tell us that. But we can say that the next frame has got nothing. And this will reduce this human workload by a huge amount. Ultimately, when the computer has really learned what an impala is and what a hartebeest is, it will reduce even further. And this is moving rapidly forward. I think Howard will be able to show you some examples at the next presentation of the computer systems filtering out the animals and giving each its best, the best shot on identification. But as I say, we need to put in a lot of information into the computer to get the answers out. Thank you very much. I think I've probably gone over time.
Thank you so much, Richard. Thank you. This was a really interesting tutorial and also a great conversation starter. We won't be able to do the questions, but we'll be able to post them on WildLabs. So if you follow through next week after the recording is up, you can ask Richard and he'll answer in the WildLabs East Africa group. So thank you, Richard, for the great presentation. And thank you everyone for joining us. We'll end here, but you can stay over for a few minutes if you want to chat with Richard. So also next week, Howard will be joining us for a session similar to this. So we'll be able to follow through the conversation and you can ask Howard more questions on the same topic. Thank you so much, everyone, and I hope to see you next week.